Right, class 12 boys, we have been discussing about different types of genes, and in that we have talked about autosomal genes, sex genes, chromosomal genes, jumping genes, transposons, holoandric genes, sex chromosome genes, number of other genes that we have talked about, constitutive genes, lethal genes, and all those. We carry a little further as far as different types of genes. Today we are going to discuss about structural genes. Structural genes are a set of genes which are present in the chromosome and which are active. That means physiologically they are active. They are responsible for synthesis of proteins or polypeptides. These structural genes, without which an organism cannot survive. If there is some defect in these genes, then that will make a major difference as far as the survival of the organism. The structural genes are responsible for synthesis of proteins or polypeptides. So these structural genes are very important for the survival of the organisms. Then we have the regulated genes. Regulated genes is the other type of gene whose expression of a gene which is controlled by the other gene. That means regulated means it is regulated by somebody. It is regulated by somebody. That means there is some other gene which regulates the activities of this particular gene. The expression of this gene is regulated by some other gene. Such a gene is said to be regulated. That means it cannot work on its own. It has to have a gene which is known as the regulator. Then we have the operator gene. Operator gene is very important as far as the functioning of the structural gene is concerned. It controls the function of the structural genes. The operator gene controls the function of the structural genes. That means when should the genes be switched on or when it should be switched off. That is the function of the operator gene. Then we have the next type of gene which is called the promoter gene. Promoter, as the name suggests. It promotes the process of transcription by providing a space where the RNA polymerase is attached. That means whenever transcription takes place, that means from DNA to RNA, whenever the message is converted from DNA to RNA, that particular message, that conversion is called transcription. So for this transcription, conversion of message from DNA to RNA, a site is required where the RNA polymerase associates, then after the transcription starts. Such a point on the DNA is said to be a promoter gene. The promoter gene to which the RNA polymerase is attached is required for transcription. Then we have the operant gene. Operant gene is a cluster of genes. That means there are a number of genes together which are packed together and they are transcribed together as well. They are transcribed together as well and their function is also almost acts simultaneously. That means it is a sort of a gene which controls the functions of various enzymes, or we can say certain substances that is required for a particular metabolic activity. Take for example the metabolism of glucose. It starts from glucose and ends in carbon dioxide. So this particular process by which the glucose is finally broken down into carbon dioxide and water plus 38 moles of ATP, that particular process requires number of enzymes. These enzymes are produced by the polycistronic mRNA. Polycistronic means there are a number of cistrons which are joined together to form a mRNA. And these number of cistrons are responsible for production of enzymes which is required at different intervals as far as the metabolism of glucose is concerned. Such a gene is known as operant gene. operon gene, cluster of structural gene that produce a polycistronic mRNA. That means this structural gene that is present on the DNA, they are a cluster, that means number of genes put together makes up the operon, which is responsible for formation of polycistronic mRNA, and this polycistronic mRNA is responsible for production of number of enzymes that is required. Number of enzymes that is required for breaking down. You take for example breaking down of glucose to carbon dioxide and water. Such a gene is said to be operon gene. Now we go on with the sex determination. Sex determination, which we have studied in the earlier junior classes as well. Here we'll deal a little bit in advanced stage as far as sex determination is concerned. As we know, in the case of diploid organisms, there exist a separate male and a female individuals. In the case of diploid organisms, we see there is a separate male and a separate female individuals. In the case of diploid organisms, we see there is a separate male and a separate female. Like in the case of human beings, we have women and men. They are two different separate individuals, which is produced as a result of the different genes or chromosomes which are involved in distinguishing them. So there is a mechanism. Since there is separate individuals of male and female in the diploid organism, there has to be a mechanism by which they can be distinguished or they can be produced into male and female of the species. How are they differentiated? The first factor that is responsible as far as sex determination is concerned, which we normally see in the case of reptiles, it is the temperature, the abiotic factor, the environmental basis of sex determination. The environmental basis of sex determination, which basically influences the sex of an organism by the surrounding conditions. Or we can say the abiotic factors, especially temperature. In the case of reptiles, certain degree of temperature gives rise to male and certain degree gives rise to female. So that is what we see as far as the environmental basis is concerned. Now, as far as the sex determination is concerned, there could be non-chromosomal gene also which is responsible for the determination of sex. As in human beings, which we have already studied, we have X and Y chromosomes. But there are certain organisms, especially in the case of bacteria, which are influenced by the non-chromosomal genes. That means extra chromosomal genes. Besides the genes that is present in the chromosome, which is required for the functioning of the life processes, this non-chromosomal gene, which is an extra chromosomal gene, which is present in the plasmid, extra chromosomal genes, which is present in the plasmid. These plasmids contain genes which are known as the fertility factor as well. And this fertility factor determines the sex in the case of bacteria. And as far as sex in bacteria is concerned, it is positive or negative strain. We do not have male and female, but we have positive and negative strain in the case of bacteria. And that is determined by the plasmid, which is a non-chromosomal gene, which is an extra chromosomal gene which contains the fertility factor. Now, we're moving on to the chromosomal basis of sex determination. In sexually reproducing organisms, like in the case of human beings, in the case of human beings, the sex is determined by the presence of either X or Y chromosome. Presence of or absence of X or Y chromosome. Like if it is XX, then it means it is a female. And if it is XY, then it becomes a male. Now what is that which is present in Y that determines whether it is going to be a male or a female? The Y chromosome carries a factor called the testis determining factor, which is also referred to as TDF. Y chromosome carries a factor called testis determining factor, also known as TDF. It is present on the small arm of the Y chromosome, on the small arm of the Y chromosome. This testis determining factor is responsible for producing sex determining regions of the Y chromosome, SRY. Sex determining regions of the Y chromosome or the SRY. Now, when XY an individual having X and Y carries this SRY factor there, XRY gene present in it, determining region, sex determining region on the Y chromosome, then that individual becomes a male. Take for example, SRY, sex determining factor, which is basically produced by TDF, testis determining factor, which is present in the Y chromosome. When XY, this has to be male as far as what we have studied so far. If this XY chromosome has SRY factor, then it becomes a male. If it has SRY factor, then it becomes a male. Now another XY, as far as the knowledge of plastine is concerned, this also has to be a male. But here what happens is, it does not have SRY factor. So this becomes a female. So even though an individual has X and Y chromosome, but that sex region of Y is absent, or we can say testis determining factor is absent, that XY individual also becomes a female. Now XX, no SRY factor, becomes female. But if XX has SRY region in the Y chromosome, then that person becomes a male. Obviously, they become incomplete male or incomplete female. We'll talk about that when we talk about the other part, the defects of transmission of hereditary characters. In that we'll talk about this. Now, who is responsible for determining whether a child that is born is a male or a female? This also we have discussed in our previous classes in 10, 9, maybe 8 as well. We already know that it is the father who is responsible for determining the sex of the child that is being born. Because father is heterogametic. That means father has XY chromosome. That is why when gamete formation takes place, he can produce two types of gamete, one with X and other with Y. So, but the mother is XX, so she is homogametic. During gametogenesis, she can produce only eggs with X. So now, when X of sperm of father combines with X of mother, definitely the child will be a girl child. But if father contributes Y chromosome, then it becomes XY. So what we see here is father is heterogrammatic, whereas mother is homogametic. Hetero means they can produce two different types of gametes during gametogenesis. whereas mother can produce only one type of gamete, XX. So the contribution of father is responsible as far as the determination of the sex of the child that is being born, because father can contribute either X or Y sperms. So here we need to remember that Y is present only in the male, and it is only the male, that is father, who is responsible for contributing the Y chromosome. That is why father is responsible for the sex of the child that is being born. Just the reverse of what we have discussed in the case of human beings. In the case of birds, the male is homogametic. That means male is homogametic. That is, male is ZZ, whereas female is ZW. So in this case, it is the female which determines the sex. In case of honeybee, haploid is a male, and that too a fertile male. Haploid, that means only when one set of chromosome is present, that produces a fertile male. A diploid can be a fertile queen or infertile worker. So here we can see there are three classes of bees that we see. A fertile male, which is haploid, whereas diploid can be a fertile queen or an infertile worker. In the case of grasshopper, it is XX plus 22. XX plus 22 is a female. XX plus 22 means XX is the sex chromosome and 22 is the autosomes. This produces a female. And X0 plus 22, that means there is one less X, produces a male. So this is as far as sex determination and different type of genes. We limit our discussion till this today. In the next class, we'll talk about more important topics like linkage and crossing over and all those in the next class.